Cousin Center lecture series. It's really my pleasure to introduce Monika Fleshner. I've known Monika for, I don't know, like just a couple months, right?
Yeah, forever.
We've known each other for a long time. She is really a testament to the excellence and the outstanding academic environment of the University of Colorado. She received all of her degree training at the University of Colorado, except for took her undergraduate degree. And has really gone up the ranks, is now a full professor at the University of Colorado. Has been a major force in looking at stress and also in exercise and immunological mechanisms. And along with Steve Maier and Linda Watkins, has really attracted many outstanding people to the University of Colorado and trained them as well. She has had several leadership positions at a national level, not the least of which is the president of the Psychoneuroimmunology Research Society. She's won several awards, and most importantly at the University of Colorado is the Distinguished Lectureship Award, and I think you'll be very pleased to hear her lecture today. Not to put any pressure on you.
Yeah.
I've heard her lecture before at American Psychopharmacology meeting, and she's one of a skilled lecturer. So we're really excited to hear about her current work today, looking at the microbiome and exercise.
Okay.
Welcome, Monika.
Well, thank you, and we'll hope if I can bring this back up again. Don't know what happened to my presentation. See, this is stress. It was here a moment ago. Anybody have any ideas?
There you go.
See, I brought this is the newest MacBook. Those of you, see, you can get it in gold, in rose. And I had to bring it, and then I brought the wrong one of these. So that's what happens. So anyway, I'll start off a little bit stressed out. So what I want to speak with you today is try to give you a concept of this approach of studying the differences between the impacts of stress in two different contexts, in the presence of being a physically active versus a sedentary organism. So I think it's beyond contention that daily moderate physical activity has many positive impacts on mind and body, and this can be found throughout the human and animal literatures. One of the primary emphasis or areas of interest for my group was trying to understand how regular physical activity also changes stress robustness. So I am a stress physiologist by training. I'm in the Department of Integrative Physiology, and I spend my life studying the stress response. And so terms like stress resilience and stress resistance gets tossed around, but they mean something special to me. So when I talk about an organism that's stress robust, what I mean is an organism that is both stress resistant, that is, it can endure longer, greater, or more intense stressors before experiencing negative consequences, as well as stress resilient. That is, that even if you experience a negative consequence, you bounce back faster. Okay, so stress robust is a concept that encaptures both of those. And regular physical activity promotes this stress robust phenotype. So using this knowledge that we have, I wanted to work with an animal model so I could get at some of the underlying neurobiological and physiological mechanisms of stress robustness. And how do you then change the activity status of a rat? So when I joined the Department of Integrative Physiology about 19 years ago as an assistant professor, I asked my colleagues who were exercise physiologists, how do I change physical activity in a rat? And they said, well, put them on a treadmill and have them run every day. And so my very first experiments tested the impact of running on a treadmill on the antibody response, which is what I was interested in. And I wanted to see whether running on a treadmill would prevent tail shock-induced suppression in antibody. But what I found is that making an animal run on a treadmill produced as great of a suppression as tail shock. So that wasn't going to work. So we went on to explore a little bit more and discovered that actually rodents would love to be physically active if they had the choice. So if you house an animal with a running wheel in their home cage like this, they will choose to hop on that wheel and run every night. So here's the kinds of data that we can get. We use a lot of in vivo biotelemetry data where we implant animals with telemeters so that we can measure heart rate, EEG, core body temperature, spontaneous movement with no wires. You can do this 24/7. And if you do this kind of an experiment, what you can see is this is activity in the cage, XY movement. This is the light-dark cycle. You can see a sedentary animal does indeed show a diurnal rhythmicity in his spontaneous movement, but compared to the amount of activity during the active cycle with an animal who has access to a wheel, it's far diminished. So an animal who has a wheel hops on his wheel every night and runs throughout the night. This is the actual distance. We can also monitor every wheel rotation. Now, when I first started doing these experiments, people criticized this work saying, is this some sort of a stereotypy, some sort of a abnormal compulsive behavior that these animals are doing? But actually, there was a very fun paper that came out in the Proceedings of the Royal Academy of Science where some scientists took running wheels and put them out in gardens and near dumps and had little hidden cameras, and sure enough, the wild rats hopped on the wheels and ran. I'm not kidding. This is a published paper. The little garden mice did. They even captured a frog. So this movement is something by choice, and we know that this has great impacts in changing these animals' mind as well as body. So if we just look at baseline differences between laboratory rats that live in an environment with a locked wheel versus those that have a mobile wheel, we can see activation of the reward pathways by running on the wheel. We see beautiful improvements in sleep. We see a lot of different brain adaptations, which not only my group, but many labs have reported. Changes in dendritic complexity, increases in growth factor, alterations in the primary neurotransmitter systems. In addition, peripherally, you see changes in core body temperature, increases in endurance. So people often ask me, well, how much exercise do I have to do to get the stress-robust improvements? And I say, well, that's hard to map out, the movement of a rat and a person, but I can look at it in terms of the fitness outcomes. And so my rats are not athletes. They're not super studs. They can run about 2 times longer on a treadmill than their sedentary counterparts. So we can do that. That's not super amounts of exercise. So they're not extreme, but they are regular. They need to run every day. And then, of course, there's a lot of metabolic changes that are very interesting that we won't get to talk about today because we're focusing on the brain. But most importantly, this model has allowed us to reveal how physical activity status modulates the negative impact of stress. Now, it just depends upon your view of the world. The glass is half full or half empty. You could either say that those of us that are sedentary are stress vulnerable, because this is a very abnormal state for the human being, something that has evolved very recently. Or you could flip it to the other side and say that those that are physically active are stress robust. Regardless, the experimental design is the same. Just depends upon how you want to view who's the control and who's the experimental group. General experimental approach is that our subjects are laboratory rats. We've studied males, females. We've studied them across the age span. We've studied outbred, so a lot of genetic variability, as well as inbred animals. And the fundamental design I'm going to present today is as follows. Animals come into the laboratory. They're housed with either a mobile or a locked running wheel for 6 weeks and left undisturbed. And then we expose them to an uncontrollable tail shock stressor. So this stressor comes from the world of learned helplessness and is capitalizing on what we know about what makes a stressor very negative for an organism. So it's uncontrollable, unpredictable, and randomized tail shocks. The entire stressor lasts about 90 minutes, and it's known to produce long-term negative behavioral consequences. The primary focus today is learned helplessness behaviors. So you see exaggerated freezing responses when you test these animals 24 hours later, a surrogate for anxiety, as well as a failure to solve a simple shuttle box escape task, some features of the cognitive deficits that you see with depression. And then after we do the behavioral testing, we can also we can sacrifice the animals and examine the neurocircuitry as well as the physiology. So in a nutshell, I wish I could tell you the story behind all of these different observations, because each of them have unique subserving circuitry changes that occur. But in general, what we found, and this is work focused from my laboratory, but others have done this kind of stuff as well, is the very first experiments showed that the animals that were physically active failed to show stress-induced suppression of KLH antibody. They have a modulation of sterile inflammatory responses, especially in adipose, as well as some changes in neuroinflammatory signals. So, for example, animals that are physically active show a more rapid recovery to interleukin-4, one of the inhibitors. So they show less suppression of inhibition on these inflammatory pathways, as well as CX3CR changes, they bounce back more quickly too. So the immediate response to stress is to suppress these anti-inflammatory pathways to allow the inflammatory process to occur, and a physically active animal bounces back faster. That's that resilience piece. This regular physical activity also has some really interesting effects on protecting sleep disruptions as well as diurnal rhythm disruptions produced by stress. Sympathetic nervous system constraint. So in general, what you see is that a sedentary animal responds with a stress response that's overboard. They drive it to the extreme and they don't shut it off very well. Physically active animal still has a very nice stress response, which we want, right? It's an adaptive response. But they turn it on and they shut it off. Brain, there's all kinds of changes in protection of BDNF, stress-induced reductions in BDNF and reductions in neurogenesis produced by stress. We'll focus a lot on serotonin dysregulation, and also the dopamine response is changed. And then finally, this emotional behavior protection. Animals that are physically active and get exposed to a learned helplessness stressor fail to show the classic learned helplessness behaviors. Here's the kinds of data we'll see a lot of. This is one of our first papers where we describe this. This is the exaggerated fear measurement, typical of learned helplessness. So animals that were sedentary, typical housing environments, were exposed to this tail shock stressor 24 hours later. Then they're put back into a little cage like this. You give them a little bitty foot shock and you measure their fear response. A normal, non-previously stressed animal shows a little bit of fear. An animal that was exposed to uncontrollable stress shows exaggerated fear. People call this anxiety, and it's reversed by anxiolytic drugs. 6 weeks of wheel running completely prevents it. So the input is identical. The only thing that's changed is their environment. The other test is the shuttle box escape deficits. The animal needs to learn to run back and forth through this door to shut off a foot shock. Once again, an animal that was previously exposed to uncontrollable stress fails to figure this out. He just gives up. Classic learned helplessness. Just can't figure out if I run through the door and back again, I'll terminate the foot shock. 6 weeks of wheel running completely prevents that deficit. So now we're armed with an outcome that is changed by physical activity. But what we really want to do is see, well, what's different about the way these brains respond that could subserve these behavioral differences. Luckily, we could stand on the shoulders of Steve Maier, who's done an enormous amount of work mapping out the circuitry that's specific for learned helplessness behaviors. So what we know is that if you expose an animal to uncontrollable but not controllable tail shock, you see an exaggerated dorsal raphe serotonergic response. That exaggerated dorsal raphe response leads to a downregulation in 5-HT inhibitory autoreceptors, leaving the DRN in this hyper-excitable state. 24 hours later, you give an animal a little bit of shock when you're testing them in the shuttle box escape or you're looking for the fear response. And the animal's brain responds with exaggerated serotonin release into the basolateral amygdala, which is responsible for the exaggerated fear, and the dorsal striatum, which is responsible for the shuttle box escape deficits. And he's done all the pharmacology to map this out. He also knows that a controllably shocked animal fails to show this cascade of events because of this inhibitory medial prefrontal cortex break. So we stood on the shoulders of Steve, and we wanted to see, well, what part of this circuit is changed with physically active animals? So the first thing we did was we wanted to see whether, just like a controllably stressed animal, a physically actively stressed animal shows a constraint in the DRN response to the stressor. And so this is using double immunohistochemistry. So what you see is that the brown is labeling the serotonin neurons and the dark brown is labeling FOS, a marker of neural activation. And we can do a lot of very nice neuroanatomical subspecificity that I won't bore you with today, but basically what you want to see is that non-stressed animals have very low levels of activation of these serotonin neurons. Animals that are exposed to the uncontrollable stressor show a very large exaggerated activation of these serotonin neurons, and animals that are physically active show this constraint. Not the absence of a stress response, but a constraint. So fewer neurons are being driven by exposure to the stressor. Well, what's responsible for this? Of course, the first thing you would think is this medial prefrontal cortex inhibition, right? These animals are choosing to run, right? So they have control over their environment. It seems reasonable that maybe this same circuit is supporting this constraint on the DRN. So we did a whole bunch of experiments. I really thought this was going to be the right answer, but it's not. You can lesion this medial prefrontal cortex, and it has absolutely no impact on the constraint that physically active animals show to the dorsal raphe. You can manipulate the controllability of exercise. So in this experiment, we had special wheels made that were yoked to the running animals. So every time that one animal chose to run, the other wheel turned. Now, when you start this, you had to kind of teach the animals that was coming, you had a little tumbling problem. Because the cage, they didn't figure out they had to keep moving. But once you train them a little bit, make them understand when the wheel moves, you run. I thought for sure this was going to work. The animals who had no control would show no protection, but they did. If they ran in the same pattern as the voluntary animals, both were protected. So you don't throw away your treadmill yet. They still showed stress protection. So it's not the medial prefrontal cortex. So what about intrinsic changes within the DRN and these projection sites? And what we've been able to show repeatedly is that wheel running by itself causes an upregulation in these inhibitory autoreceptors, these 5-HT1A autoreceptors in the dorsal raphe, in the subregions that are important. And so this by itself could provide a brake on the DRN during uncontrollable stress. We also have some small changes in the 5-HT2C receptors in the projection regions that might also contribute. In concert then, when you get exposed to this uncontrollable stressor, your DRN has a bigger break. You don't respond with this excessive serotonin response. If that's the case, if it's functionally true, we should be able to show that using more functional measures. And so here what we did is we wanted to see whether a physically active animal exposed to stress shows a constraint in serotonin output into one of the projection sites, like the dorsal striatum. So to do this experiment, Peter Clark, a previous postdoc in the lab, set up animals with dorsal striatal microdialysis probes, allowed them to run, did the surgery, allowed them to recover, then exposed them to the stress and collected dialysates. Remember, the idea is that if you get exposed to uncontrollable stress, you produce this sensitized dorsal raphe, so that 24 hours later, if you get a little bit of stress, you respond with a huge release of serotonin. And so, of course, it must have happened because I'm showing you the data. This is the experiment. This is in vivo microdialysis serotonin measurements in the dorsal striatum. Running per se doesn't change anything with this response. But if you were previously physically active and you get exposed to 100 tail shocks, so you have a potential sensitized response, you see here in the Sedentary animals, an exaggerated serotonin response that's constrained with physical activity. So the upregulation of 5-HT1A inhibitory autoreceptors is functionally real. So we've had a lot of fun using this animal model to untangle changes in the neurocircuitry that might subserve stress robustness. More recently, as is often true and driven by the students you bring into the lab, one of my doctoral students was very interested in a developmental component of this effect. Does it make a difference if you start exercising when you're younger versus later? So her first experiment was to test full-blown, not aged, but middle-aged adults in the rat world versus juveniles, which are postnatal day 24. They're not quite babies, but they're not too far off. And we let them live either with a running wheel or not for the typical 6 weeks. We exposed animals to the tail shock stressor, and then we tested them immediately. And we were also very interested in the persistence of the effect. Gosh, do we have to really exercise every single day? Like I'm traveling, I'm not exercising. I can feel my stress robustness slipping away. So do we have to do it every day, and how much of a window of cushion do we have? And so we tested with the wheels locked after 15 days, and we tested with the wheels locked after 25 days. So first of all, the animals overall run about the same, the young animals and the older animals, because these guys aren't ancient, they're just grown up. And here's what we found. So if we want to look first at the exaggerated fear, the anxiety measure, you can see the sedentary animals that are exposed to the stressor 24 hours later show this exaggerated fear response in the adults as well as in the juveniles. If you run on your wheels, you're protected if you're an adult immediately, but by gosh, it's already gone 15 days later and 25 days later. So you have to keep on running, okay, in the adult situation. Importantly, however, and very exciting for us, is that the young animals who began running when they were just postnatal day 24 were protected immediately, but protected at 15 and 25 days of no running. So they have a very long-lasting change. Now, does that also hold true for the shuttle box escape? Pattern looks very much the same, right? You're adults, you have this failure to learn, so your stress causes this depressive-like behavior. Same is true in the juveniles. You're protected immediately after running, but once again, not at 15 or 25, but your protection persists if you're young. So we began to think, that's pretty wild, 25 days, and we haven't gone out farther yet. But you produced a real change in the phenotype of this animal so they can endure this stressor and have this protective effect last much longer. Oh, sorry, that's my shuttle box test again. So we wanted to see whether the primary change in the brain in the dorsal raphe also occurred in these animals, and if that persisted as well. So that's that upregulation in 5-HT1A, that break. And so we repeated the same kind of an experiment. We have adults and juveniles running on wheels for 6 weeks. We're then measuring the 5-HT1A receptor expression immediately after stopping running and 25 days after stopping running. And we're looking at the dorsal region of the dorsal raphe, rostral, mid, and caudal. So these two regions are your primary targets. And what you can see is that the adults and the juveniles have a little upregulation at the immediate time point, but more impressively is this 25-day time point. 25 days of no running, it's completely gone in the adults, but it persists in the juveniles. Here again, 25 days after running has stopped, you have this persistent upregulation. So you've got something that looks almost permanent. And there are others who are really diving into the potential epigenetic changes that occur with wheel running in different parts of the brain. But what could be responsible for this? Oops, I lost my titles. So I did a conversion from PC to Mac, and there used to be a title up here. Let's see if I can remember what the title exactly was. So we know that we've got a long-term persistent effect with juvenile onset running. Could the gut microbiota be important? That transition was a little rough. And now my bacteria, that title is gone too. So what do we know about the gut microbiota? I'm sure you guys have seen this around. It's sort of the hot new contribution to thinking about physiology as a whole. We know that these commensal symbiotic bacteria that colonize our intestine in this beautiful carpet of bacteria are adding enormously to the diversity of our physiology. The gut microbiota are the microbial contributions. The gut microbiome are all the genes that come with that, both microbial as well as viral. And there are millions of these microorganisms residing in our intestines, 10 times more bacteria than cells, yada, 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 right? So they're contributing to a lot of diversity in our physiology. Interestingly, there is a growing, exploding literature, this is already way out of date, showing that particular species of bacteria seem especially relevant to brain and mood changes. So, for example, the lactobacillus species, specifically Lactobacillus rhamnosus, as well as bifido, are all clustered together as being especially important in mood-altering effects, decreasing anxiety, as well as potentially this protection against the adverse consequences of stress. In addition, if you feed, so in a probiotic fashion, feed animals, and there's some work in humans too, but this is an animal study, Bifidobacterium infantis will cause changes in serotonin metabolism as well. But even more interestingly for this project are the potential metabolites that are made from some of these types of bacteria in the gut. And so one of the important contributions to digestion, especially of certain sugars and starches that are resistant to typical digestion, are a class of bacteria that produce short-chain fatty acids. I'm going to talk about one metabolite in particular, butyrate, because butyrate also is a histone hyperacetylation, so a histone deacetylase inhibitor. So it is involved in epigenetic changes. So you've got this change, butyrate induces histone hyperacetylation in bulk chromatin within the hippocampus. It also increases this BDNF promoter acetylation. There's a lot of additional work that's going on beyond this. Butyrate also can attenuate some of the decreases involved with BDNF protein expression. So it does a lot of these interesting potentially contributing changes in the brain that might play a role in some of the long-lasting changes that you see. Now, the gut-to-brain communication schema sort of looks like our beginning PNI. You guys remember our PNI little diagrams that we all had? As how is it that the nervous system communicates with the immune system, vice versa? This is the same level that we're at right now for trying to understand how things that are happening in the intestine and consequences of the microbes and the metabolites in the intestine could communicate with the brain. So we have multiple pathways, and our short-chain fatty acids are always ones that are considered. In addition, this is screwed up too, but in addition, interestingly, the gut microbial community structure is more plastic when we're younger. So if you look here, these are data actually from my lab where we measured a measure of microbial diversity. So this is Shannon entropy as well as the number of species. So what we're talking about when people say alpha diversity, they're talking about the numbers of different types of bacteria that are within the intestine. So sort of the richness and diversity of your forest. And if you think about that, that can be advantageous from the perspective of, let's say you need, bacteria X to digest some particular new plant food that you had. You'll have a better chance at successfully digesting that if you have a larger diversity of types of bacteria available to expand to respond to that new food that you've introduced. Okay, so it just gives you more options. And as you get older, your diversity increases across time. And interestingly, early life exercise seems to have a greater effect in younger rather than older animals. So here's just looking at microbial DNA, a rough, crude measure of gut microbiota density, and you can see previous to wheel running, the animals, though this is after 6 weeks of, no, this is only 7 days of wheel running. So 7 days of wheel running, you don't see much of an effect in the older animals. If you take the wheel running away, you also don't see much of an effect, but your juveniles respond very rapidly and they have a persistent increase. In addition, we did a large experiment where we did full-blown 16S analysis, of which we won't talk much about today, but it is published in this +1, where we compared the impact of adult onset exercise with juvenile onset exercise. And we measured it, at the phyla level, and then we broke it down into much more resolution. And what you can see is that the adult onset, this is after 3 days of running, 6 weeks of running, and then 25 days with the wheels locked. You can see little changes that are happening here with the adult onset, but it's much larger in the younger animals, and it persists. And this change could be indicative of what you'd call a lean promoting ratio between the Bacteroidetes and Firmicutes. But for the importance for this presentation is simply the more dynamic regulation of the response. You can also look at classic beta diversity differences across time. You can see that the juvenile animals and the sedentary animals sort themselves much more distinctly into populations than the older animals. So in general, the younger gut microbiota responds to the exercise stimulus with a greater and more rapid change. We can also look at some of the actual mood altering. This is still using 16S analysis for lactobacillus species. The young animals show this dynamic change. The old animals show very little detectable change. These are butyrate producers. Once again, your young animals show these dynamic changes. The old animals show very few changes. In addition, we can use some more old school selective culture techniques. So here we're using culture techniques that allow us to identify specifically Lactobacillus rhamnosus as well as bifido. And you can see that you get this nice dynamic change in the younger animals. And if we actually look at short-chain fatty acid fecal concentrations, a surrogate for the fecal butyrate, commensal bacteria butyrate concentrations, you can see that the juvenile animals show a much greater increase than the adult animals. So the idea, it's just in its infancy, but there is some suggestion that these young animals show a faster response to the exercise stimulus and a more persistent response. And that occurs both in the brain as well as in the gut microbiota. So maybe these metabolites like butyrate could be contributing especially to that long-lasting persistent effect. And so like I said, we're just starting this. We did do one experiment to kind of try to get at that. Young animals not only show a more persistent effect of exercise, but they show it in half the time. So if you take an adult and let him run for 3 weeks, he doesn't have this stress-robust phenotype. If you take a juvenile and let him run for 3 weeks, he does. So he gets it faster as well as prolonged. Typical superstar young stuff, right? You're just more plastic and more dynamic. So in this experiment, what we did is we took young animals, we let them run for 3 weeks, then we locked their wheels, and we tested them 3 weeks late, 15 days later, yeah. And then in this experiment, what we did is we examined the impact of giving animals antibiotics throughout this whole period of time versus not. So the oral antibiotic dose, we were able to show, here's the impact it has on fecal butyrate, completely obliterates the fecal butyrate effect produced by running, and it also completely prevents the protection. So this is the stress protection that you see 15 days after locking the wheels in the young animals. See, they failed to show your shuttle box escape deficit. If you gave them antibiotics during this time, they also failed to show the long-lasting effect. So we have a lot more work to do. But taking out the gut microbiota during this 3 weeks, remember the wheels are locked, no more antibiotic. You test them 15 days later, and these animals no longer show a persistent protection, whereas these animals did. These animals also don't show the butyrate change. So maybe the butyrate change is contributing to that long-lasting, more epigenetic change in the 5-HT1A upregulation in the dorsal raphe. And so that's kind of where we're going to go with this in the future. So, of course, a lot more work has to be done, but that doesn't prevent us from writing a review paper like everybody else does before they really know what's going on. And so this is our speculation, that there's something about exercise that changes our gut microbial species to promote these mood protective bacteria, as well as promoting these butyrate types of producers. They have a lot of different impacts and a lot of different potential signaling routes, either potentially through the vagus, maybe the circulation, although, butyrate getting out in the circulation in high concentrations is going to be a difficult task to prove, but we'll see. And in the end, they may be contributing to the impacts that exercise has on stress robustness. And so with that, if I had my PC, these guys would wave. I mean, if I had my Mac, they would all say hi. This is my group that helped with this work. And Aggie, this is Aggie Mika. A lot of this work is her dissertation research. And I'll take questions from Boulder. Thanks. Yes?
Is that do you use a broad spectrum antibiotic?
Yes, broad spectrum. Yes, exactly. Yeah, and I could have shown you more of the characterization of the impact that it has. Of course, it doesn't take it to zero, but it does take out. It takes out the bacteria that we're especially interested in. I can't find anything that's more targeted. I would prefer to do something more targeted. She's smiling like everybody else. I've heard of several creative approaches that people are doing to try to do more targeted elimination. They're trying to stimulate the immune system against targeted bacteria, but of course, the mucosal immune system is targeted toward quiescence and not making immune responses, so they're having real challenges there. But that's a neat idea. Anyway, so yes. That's a question. Answer the question. Yes, sir.
If you co-house the ones which run with the ones which are sedentary, which wins?
Well, I haven't done that experiment, but that is exactly the kind of experiment I want to do, and I want to do it very early. And I've tossed around multiple ways to do this. So do you start with animals that are notobiotic and match them? So you have an exercise with a previous notobiotic, so you have the greatest possibility of creating the most similar gut microbiota. Do you do it with animals that you've given an oral antibiotic to reduce it and then cohabitate, or do you just wing it, and hope that they drift? Because what will happen a lot of times is that they don't. Who wins? They end up sort of drifting. Yeah, they have a drift in the middle. Yeah, it's a great experiment. So I'm hoping I can get some more funding to be able to do those kinds of studies, because there's a lot of stuff you could do that's not so extreme, that would still get at the question.
Also, have you seen the 5-HT receptor modulation when you put the antibiotics in?
We didn't do that yet. So that's, of course, the next follow-up experiment to see whether this blocked the 5-HT1A receptor, especially the persistent effect. That's what I'm kind of hoping, but I don't know that yet. Up in front, and then I'll go to you. Yeah.
What was the rationale behind using postnatal day 24, and do you think there's a critical developmental window that you're tapping into?
Well, the rationale was in part practical. So that is the earliest that we can order animals in without having to order the dams too. So it was literally just post-weaning. And also we have some additional other work using prebiotic diets. We also wanted them post-weaning then, so we had a match up there. So most of it was really practical rather than fully connected into the developmental changes that are happening. But certainly we are still capturing a lot of dynamic changes at postnatal day 24. Yes.
What does your running intervention do to their sleep? They're running at night a lot, it looks like.
Yeah. So what you see is, it's kind of cool. What you see is that the running, well, during their night, and they sleep during their day, right? So their sleep during their day, you see more consolidated sleep. So you see longer REM bouts, which in a rat, are much shorter than a person. They're little bitty spurts, as well as NREM bouts. But even more profound is really the stress disruption. So when you expose these animals to a stressor, you see a lot of changes in sleep, including this sort of fragmentation. And the animals who got to run before that stressor show less of that negative effect of fragmentation.
How much of the effects of this exercise do you think are shared by the impact on sleep that you're getting? Or can you make that determination?
No, I can't, but I'm surrounded by sleep researchers in integrative physiology, and I've got a grant with one of them, and we're definitely going to try to. That's always a tricky thing, but it's definitely something that we're interested in. I was surprised. We also have another paper that's in review right now looking at prebiotic diet and changing sleep. And there you see really interesting changes, but once again, it's the young animals, not so much the older animals. And of course, I don't have all the data to support this speculation, so forgive me. Don't reject the paper if you're reviewing it. But it looks like, and I don't know if this is even good, but it looks like if you look at the developmental changes in sleep in these animals across time, it looks like the animals that are put on the prebiotic diet move more quickly through the developmental changes. Now, is that good or bad? I don't know. I really don't know, but it seems to accelerate this change in their REM and not REM oscillations as they develop a more adult-looking sleep pattern. And I'm not doing justice to it because there's a lot of different sleep measures in there that get at more of the subtleties of it. But yeah, it does, and it has a real effect. It really does. And I don't know why. Yes, here.
Is your idea that the juvenile mice have a more prolonged effect because their microbiota is more plastic than the adult microbiota?
Well, that's one idea.
And if the data that you showed with the antibiotic experiment, you showed with the juvenile mice, have you done that with the adult mice and show whether there's...
Not yet. We're working on it. We've done a series of experiments, several of which had a design confound that my graduate student just didn't think about, like that 1 week versus 3 weeks of oral antibiotics might be something to be concerned about controlling for. So I've actually done the experiment, but not in a way that I can really interpret the results, because she's got this problem. It's my problem too. Yes.
So it's also possible that exercise would have an impact on the pain perception, so that the exercise group is receiving less stress in the
Yes.
first place.
I'm glad. So that was one of our very, very first control experiments that we had to do to show that the input was the same. Now, how do you do that experiment? I can tell you what we did. What do you think you should do?
Measure their skin conductivity.
Yeah, you could do something like that. What we did was we looked at the incoming, the periaqueductal gray, the incoming pain region that senses the pain input. And we documented that both animals had the same amount of PAG activation, suggesting the input was the same. In addition, a lot of other things are the same. Their glucocorticoid response is identical to this stressor. A lot of the peripheral cytokines that they make get a huge inflammatory and chemokine response in the blood. Most of those are unchanged. So there's a lot of reasons to believe the input is the same. But that's a great question.
So any research done on the germ-free mice or rats in wheel running and stress response?
Have I seen germ-free with wheel running? I can't come up with an experiment off the top of my head. I don't like the germ-free model. just because those animals are so aberrant developmentally. But does anybody know? I'll have to search that because it's bugging me. But I don't remember exactly that experiment. There are other people who have done work with probiotics in wheel running, but I don't know of anybody who's actually taken a germ-free and looked at their wheel running. Mm-hmm.
As you know, maybe the intestinal permeability is very much increased in developing rats up to day 30.
Mm-hmm.
So how much this difference in intestinal permeability could feed your hypothesis about the role of the microbiota being much more capable of influence this gut-brain circuitry?
Oh, that's great. So I've the intestinal permeability differences I viewed originally as a negative because I also had this problem with the potential, like the gut leaky hypothesis, that you can get some of these endogenous bacteria and LPS in the circulation that trigger an inflammatory process. But I don't have any evidence that that's happening. But I like the idea of the integrity difference. Maybe that would help with my problem that I have with getting butyrate at high enough concentrations in the circulation to get to the brain. That's good. Yeah, good. I'll keep that in mind. But then my prebiotics improve intestinal integrity, so that I'll have to think about that because we are combining them too. Thanks. Anybody else?
Does it matter when you do the stressor? Does it matter if you do it in the light or without part of the cycle when you do the stressor?
Yeah, other people have done that. I have not. Because the classic learned helplessness experiment was always done where you give the stressor at the very earliest part. So you're thinking about sleep disruptions again? Yeah, exactly. I know. I agree. But we haven't actually checked. That would be a really interesting experiment to do, to see if the exercise effect changed. In other words, do you have to have them being stressed during the day, so waking them up, to see the effect? I think Steve Maier has done he has demonstrated you get the effect either in the day or night, but I'll double check that. That's a great idea too. See, that's why I come to these talks. Yes?
This is kind of a naive question, but how do you think exercise-induced ketosis factors into this model?
Well, I've been trying to take a little slightly different question, sort of where you're going, is I've been trying to figure out what signal about exercise would change the bacterial populations. And so there's a few ideas. There's this whole metabolic component to what's happening with the gut microbiota and the energetic demands of wheel running and how those changes might be to provide more available, usable energy right now, okay, from a very simpleton perspective. The other thing I was thinking about was just really simple pH physiology, right? So there is some evidence that exercise can reduce the pH of the gut. And especially these lactobacillus species grow better in a lower pH. So you might have something as simple as that, sort of kicking the whole thing off. But people are working on that, trying to understand what the signals are. And the question I would have is it advantageous? So like I said, I didn't get to show you sort of the metabolic differences between early life exercise and adult onset exercise, but there's some changes that happen in lean versus adipose that would kind of feed into that argument that what's happening with the gut microbiota are helping to support this lean mass growth and less of a drive towards storage, toward adipose growth. So it's kind of cool. If you dig into that literature, it looks feasible.
Monika, since this is the PNI lecture, can you kind of bring this model around to some of the immunology work that you've been doing with your information? Because we know that there's a lot of effects of exercise upon inflammatory responses. Many outcomes that you've been looking at seem to really overlapping effects as well as exercise. So
Well,
so your slides, you have the IL-10 up there.
Yeah. So
where you're going with that.
So we've done a little bit of work. So so I've just started to collaborate with Linda on some of her neuropathic pain models, and we're getting incredible effects with exercise on preventing the chronic development of neuropathic pain. So that's an immunologically mediated event. In the brain, I've done only a little bit of work, which I showed you just a smidgen of. We did this experiment, so what I call my exploratory experiment, where we compared animals that ran for 6 weeks versus not, weren't exposed to this stressor, and then sacrificed immediately and 2 hours later. And we did laser capture of the dorsal raphe, and then we did a big gene microarray and looked for every gene that a rat has and looked at how they changed. And that was the work where we showed changes in interleukin-4 as well as the fractalkine receptor. But overall, I'm not seeing huge differences. It could be in part the intensity of the stressor. So we have done some work looking at the sterile inflammatory response in physically active versus sedentary lean animals, and looked in their adipose and showed sort of a dampening of that response. So there are immunomodulatory effects, but this model's not perfectly designed to really capture them, because I don't really have a big pro-inflammatory context, we did some work where we actually checked on their bacterial response. So this acute stress response is both good and bad. And from a positive end, animals who are exposed to this stressor and then challenged with subcutaneous bacteria actually kill and clear their bacteria faster than non-stressed animals. So we wanted to see what physically active animals who were stressed would do. Now, if you would think about it from a simple anti-inflammatory effect, you might think that they have less of the positive side of stress, but they actually showed even faster killing and clearance. So I think it's complicated. I think it's not going to be a simple dampening of the response.
I have one more question. Sex differences.
Yeah.
So,
Okay, I know I have to study it.
I'm just so intrigued by what's happening in the literature and how...
I know.
When you look at female versus male humans, you have really striking differences, and I assume that's also the case.
Yeah, and it's going to be the case, and we're going to have some. These are all males. Females run a lot more, so we're going to have some. I'm going to have some issues in trying to figure out whether it's distances versus male-female differences.
So embedded in that question is also this difference of sort of aggregated things, and I wondered, have you looked at treated things as individual data points and looked at individual variability and how that relates to some of these outcomes?
Yeah, so science is funny because it goes kind of full circle. So when we started, we used outbred animals, Sprague dollies, because we wanted genetic variability, and then we could do individual differences. And actually, we're doing some fun work right now where if you take a big group of Sprague dollies, which is kind of like us, and you put them in their home cages with wheels, they will naturally have different propensities to choose to run. Just fall out. You'll have a group that really loves it, and they run a lot. You'll have a group that's in the middle, and you'll have a group that never increases the amount of running that they do across. They do not seem to care. So we're very interested. Actually, the experiment was designed to figure out how to make these that didn't care. Haven't figured that out yet. We tried caffeine. And what happened was give them the poop. That'll be down the line. Give them some poop. Instead, we gave them a little bit of caffeine, and what happened was that the high runners ran even more. Right? The middle moved a little, but the high ones are the ones that were most. It didn't work out at all. But anyway, we're going to be able to go back and do some of that. But then I shifted gears because everybody poo-pooed Sprague dollies and outbred animals, and you had to have genetic control. So we went to an inbred, and then you have very little variability. You can see. I didn't show a lot of wheel running, but they have a pattern. It's very interesting. It's sort of like addictive behavior. They start low. Right? And you can see it ramping up. Remember, they show dopamine activation. We show the reward pathway being activated. So it's as if it's rewarding. And there's actually a whole literature out there right now on the endocannabinoid system and its role in this behavior. So So that'll be that's actually where I'm hoping to go with my low runners, is to see if I can tickle the endocannabinoids to make them like running more. If they like it more, they would do it more. But the individual differences, we can do some of that. A lot of our effects look more to be associated with persistence of running up to a certain point. The really low runners are not getting much of anything, but it's more about how persistent you are every single day rather than how far you run. So the correlations with distance per se for a lot of the measures are not impressive.
Great. Well, thank you very much.
You're welcome. Thank you for the good questions.